Bob, and hello everybody. Bob, can I just check that the laser pointer is moving and the first slide
It is,
is
and we're looking at the title in the first slide. Perfect.
excellent? Okay, thank you. Okay, so we specialize in the development of enabling technologies that improve the growth of cells in vitro, with the aim of recreating a more natural three-dimensional environment to enable us to simulate the generation of real tissue, so tissue equivalents. So the idea being that we would be able to produce genuine 3D cell and tissue architecture, which is more representative of real human tissues, with enhanced structure and function, that in turn will enable us to develop improved in vitro assays with greater predictive accuracy. So we use this technology to bioengineer tissues in the laboratory environment, and this slide shows 4 examples of a skin model, a respiratory model of the nasal mucosa, an intestinal tissue model, and an oral mucosa model. So that's the lining of the mouth. And each of those models have been developed using the technology that I'm going to be talking to you about today. The key features of these models are that we generate them following anatomical and physiological principles, such as structure is always related to function. So the structure of an epithelium, for example, is of a particular architecture for good reason, and I will explain this further when we come to this point. All of these models are generated on a physical scaffold, enabling three-dimensional cell growth, and I will introduce that to you shortly. We also design and manufacture our own plasticware for cell culture purposes, and this is necessary to maximize the ability to generate these models, as I will explain. Each of the models standing behind these models is a refined standard operating procedure, so a set of methods that have been optimized and refined. Each of the models has had in-depth characterization in terms of their structure and function. They are reproducible and robust. We compare these models to real human tissues in health and disease, depending on the nature of the model. Also standing behind these models are extensive sets of data, application notes, white papers, scientific publications. As we go through this presentation, you will see that these technologies can be analyzed using a whole range of different endpoint assays. And ultimately, the other point I'd like to make is that when we are working with our customers, we listen to their needs and we developed tailored solutions important in product development for those clients. So this is important because these models can lead to advances in research and discovery. They can improve efficiency, saving time and money, and of course there are alternatives to using animals in research. And such systems are relevant to the majority of scientists culturing mammalian cells and tissues, and there are applications for basic research, drug discovery, and product development, as I will demonstrate. So let me introduce you to the core technology that facilitates the growth of cells in 3D. So we've produced a three-dimensional polystyrene scaffold, which is presented as a 200 micron thick membrane, and you can see images of that at the bottom in profile. Essentially what happens is that the cells come into contact with this material, they go inside and on the surface, and rather than flatten out as in a 2D culture, they retain their 3D structure and they interact with their neighboring cells in a three-dimensional environment. And this allows the cells to build up three-dimensional structures. If you look at the bottom right image, you'll see that this scaffold, you cannot see the actual structure of the material because it's now full of cells. So essentially what we've produced here is a 200 micron thick slab of tissue-like material. So that's the basis of the scaffold. And then the scaffold itself is presented in a series of plates and inserts. And these are the commercial products. The brand name of this product is Alvetex. It's available from REPROCELL, and that's the REPROCELL website here. We have standing behind this technology a very large amount of applications. There are over 175 publications now from our customers who've independently used this technology and then published their outcomes. It is an established brand in the field, and it's probably now recognized as the market leader in scaffold technology for 3D cell culture as a form of physical scaffold support. But whilst it's used heavily for the concept of 3D culture itself, more recently we've been using it to bioengineer human tissue models, so to increase the complexity of our 3D systems. And to do that, we've generated a new range of exciting novel plasticware. This is now called Alvetex Advanced. And on this slide, what we're showing you are some examples of these products. So this is bespoke cultureware for customized applications, promoting the longevity of the model and their reproducibility. They are designed for improved handling and access to the model's surface, particularly important if you're growing skin and you wish to interact with the surface of the skin model at the air interface. Some of these particular formats have been designed so they are compatible with existing testing devices used in the clinic. And I'll show an example of that shortly. And the whole concept is designed so that it improves the predictive accuracy of tests to reduce the cost in the design and application of clinical trials. So in other words, the data that you're getting from these in vitro model systems is of real value and can help make subsequent decisions on the progression of a product, or actually using the data to help promote the product as well. Excuse me. So this is an exploded diagram, a schematic showing Alvetex Advanced. So this is the new technology I mentioned a moment ago. It's a very flexible, versatile, modular system. So if you just look at the middle diagram here where the laser pointer is, you'll see an exploded diagram of the insert. This is the Alvetex scaffold, and it's sandwiched and held within this insert, this cup arrangement. And that cup then can be placed in these handle holders, and they can then be suspended into trays or individual wells in a multi-well plate, or these inserts can be placed into another holder, which is a 6-station version, which holds all 6 models together, and that can also sit in a plate or a single well tray. So the whole point of doing this is to cater for alternative experimental designs and the needs of our customers. We can also control media volumes. They are designed, as I mentioned, for ease of topical application to the surface of the model. We've also carefully designed the perimeter of the insert where it houses the scaffold to ensure a leak-free barrier, so that the barrier properties are through the actual biology in the model, not through a leak around the edge of the scaffold. And we've also, as I've mentioned, created designs for non-invasive functional readouts. So this means maintaining a model in the incubator, taking it out, taking a reading, and then putting it back for another period of time, and then repeating the process. So you can do chronic studies on live materials. So for the action of a compound over time, for example. These are the inserts again, as shown in this photograph, and what we see in the photograph on the right is one of the devices where we are interacting with the surface of the model. This device actually is measuring water loss from the surface of skin. That's used commonly in the cosmetics industry on real skin during clinical trials. So that device is an example of a measuring procedure which actually is also adapted in our model system. And there are other examples as well. There's specialized pH meters. There's also systems for measuring skin tone, which I also show later. In this slide, you can see very clearly the three different handle options. And as you can see, there are different heights presenting the insert to different depths within the well or tray. Now, this is important because this enables the user to have different volumes of media above and below the insert and the model as needed. So it adds versatility. And if you combine the handles with the different trays, so the trays here, you see them all three listed, 50, 75, 125 mil, then you have a lot of versatility. You may well think that those the volumes of media are high, but remember you have potentially 6 models in, say, 50 mils, which is less than 10 mils per model. And remember, a model is not like a conventional 2D culture. It is multiple layers of many cells, and you are trying to maintain the viability of a tissue equivalent over multiple weeks. So to be able to do that, you need to think about how the regime by which we're going to maintain those models, minimizing the number of times that one have to interact with them for media changes, etc., etc. All of that is taken into account in design. So what do we use this technology for? So let's look at a couple of applications, and we're going to today look at 2 applications in relation to the bioengineering of epithelial tissues. So an epithelium, by definition, sits on the basement membrane supported by mucosal tissue, primarily fibroblasts producing extracellular matrix, along with other cell types within that submucosal compartment. The structure of epithelia are linked to their function, as I already mentioned. And if we look at, say, for example, in the intestine, we have a simple columnar epithelium designed, of course, for absorption and secretion, whereas in the skin, we have a stratified keratinized epithelium designed for protection, a waterproof barrier, for example. So they're very different in terms of their anatomy and their physiology, their function. So what I'm going to do now is show how we use our technology to bioengineer these tissues. The concept is the same, no matter which tissue that we are developing. The idea being is that the fibroblasts from the tissue are first of all cultured in the Alvetex, and during that time they produce extracellular matrix. So it's human-derived ECM, which is produced and remodeled by those cells. That takes place over time, and then at a suitable point in maturation, we then seed onto the surface the epithelium, and depending upon the type of tissue that model we're making, we may or may not raise it to an air-liquid interface to enhance differentiation. So that's the concept. Let's look then at, first of all, some examples. So we'll look first at skin and what we've been doing in this space. So skin, if we look at it in simple terms, epidermis and dermis, the dermis having fibroblasts and complex ECM, as I already mentioned, other cell types too. The epidermis itself is composed of different sublayers at different stages of differentiation of those keratinocytes moving to the surface and forming the stratum corneum. We're also actually developing models with the hypodermis as well. So first, to make the dermal compartment, we take the Alvetex membrane in our insert. We then seed into that human primary dermal fibroblasts, and we maintain them for 3 to 4 weeks. And during that time, the cells grow inside and on the surface of the Alvetex, and they populate it and they produce their own endogenous ECM. So these images on the left here show immunostaining for collagen 1, collagen 3, and fibronectin deposited inside the material. And the central image here shows a fibroblast in, this is a section of a transmission electron micrograph, and surrounding that cell you can see very prominently the fibrils of collagen. Not only collagen, we know from our proteomic analyses that there are other types of extracellular matrix present, and also factors and cytokines, all of which create a rich environment to support the overlying, in this case, epidermis. So we seed onto the surface of the dermal compartment epidermal keratinocytes, again, primary human cells, and raised to the air-liquid interface. And over a period of a few days, we produce a differentiated epidermis with all those sublayers from the stratum basale through to the surface stratum corneum. This is robust and reproducible, and we can maintain this for several weeks for longevity and recovery studies as well. We produce quite large pieces of this material, showing here, and we characterize the material, the model, in comparison to real tissue. So these images represent immunostains for many different types of marker, which are expressed in normal skin, and we cross-compare real tissue with the model, looking for expression patterns and showing that they are indeed very similar. And this is all published work as well. We also have conducted scanning and transmission electron microscopy, looking at the anatomy of the models, demonstrating the formation of the hemidesmosomes, the basement membrane, the desmosomes, the corneodesmosomes, the lamellar bodies, etc. So now we have our skin platform, we can now start to introduce complexity. So we can introduce cells that enable us to create models of aged skin. We can introduce neurons, immune cells, pigmented cells, and I shall share with you some examples of this. And these models then can therefore have specific applications. For example, for skincare product development, testing topical actives, skin exfoliation, shave care, grooming, mechanical, physical challenges such as wounding, exposure to UV radiation, sun damage, etc. And use of this bespoke plasticware compatible with clinical testing devices enables us to make appropriate recordings as well. So to produce a model of human age skin, we can do two things. The first is to utilize cells from young and old donors. So on the left, we have a young skin model from young material. whereas this is aged material, and you notice immediately the difference in the thickness of the epidermis, which is what happens when you age. You have a thinner epidermis, and there's less collagen and other extracellular matrix components in the aged model, which also happens during aged skin. As tissues age in general, they accumulate senescent cells, and we can add senescent cells into our models artificially. So we take fibroblasts, we can irradiate them, or we can treat them with chemicals to render them senescent. We then incorporate them into in this case, these are fibroblasts incorporated into the dermis at different percentages, so 0, 5, 10, and 20%. And then we can study the impact of those senescent cells on the generation of the skin model itself. And you can see thinning of the epidermis as the percentage of senescent cells increases. This is through the bystander effect and release of the SASP from the senescent cells, and that leads then to the development of senolytics and xenomorphic treatments, which are a new area of cosmetics used for aging skin. So you can use this platform for those exploring and investigating those particular types of product. We can also bioengineer human skin through the incorporation of primary melanocytes into the models. So these cells, they self-organize into the basal region of the epidermis. So this is the model here showing the dendrites of a melanocyte in a pigmented model versus the absence of them in a non-pigmented model. You'll notice this structure. This is a perinuclear cap appearing above the nucleus of a keratinocyte, the arrow showing that. And this is what happens in real skin. You can see the perinuclear caps, or the supranuclear caps, I should say, sorry, above the model, above the nuclei in the real skin, but also in the model. And this acts as a protection from the nucleic acid from the UV radiation. We can construct these models using melanocytes from different ethnicities to create a skin palette. And this is of value because then one can begin to explore the differences in skin tone and also generate experiments for testing products towards different groups of the population as appropriate. What's interesting is if you look very closely about the way in which the models become pigmented, we can follow the process of melanogenesis through its various stages. You see the 4 stages of the melanocore and the transfer of melanin from the dendrites to the keratinocytes. All this is happening in the model to produce the pigmented skin equivalent. These data show the importance of co-culture. So if we produce a epidermal only model and incorporate melanocytes, we do not see the formation of the supranuclear caps. This only occurs in the presence of the dermal compartment. So the crosstalk between the epidermis and the dermis is very important for the normal differentiation of the tissue. And that's one of the reasons why a full thickness system is ideal for studying these skin pigmentation aspects. So what do we do with these models? Well, here's an example. This is an example of using a well-known molecule which is known to lighten skin tone. So kojic acid inhibits tyrosinase rate-limiting enzyme in melanogenesis, and as you increase the concentration of kojic acid, you see the models begin to lighten. And of course, you can measure this very easily. using the skin tone device, which is also compatible with the approach in the clinic. So skin lightening is achievable, as is skin darkening, and we can do that using UV light. In this case, we've got here the UV irradiation machine, the BioSun. This is the skin color catch, the device which is used to measure the ITA and melanin index. So that's the device used in the clinic, and that's compatible with these pigmented human skin equivalents. You'll see the operator here on the right adding to the surface of the model, in this case a sunscreen, and we can show very nicely in the absence of the sunscreen, in the presence of UV, we get blisters forming, whereas with the sunscreen present and UV, you see the protective effects of the sunscreen. So that's an example of an assay whereby one can screen formulations of these products. These models are robust, and you can interfere with them from the surface. In this case, we've got an example of tape stripping. So this is a process which is often used in the clinic to collect the stratum corneum, and we can tape strip down to the glistening layer, just like in the clinic as well. As you can see from the data at the bottom here, we've got this is after 0 hours of tape strip, where after 4 days of the tape strip, you see that the model has recovered. So it's a recovery process which happens, and you can continually do this as well. Some of our work has actually used this as a surrogate for shaving. I'll come on to some shaving work in a second. These are the data showing that the exfoliation by tape stripping leads to an increase in tools. This is water loss on the surface of the skin as the barrier is compromised. But note after 4 days how the tool rebounds just as the structure is restored. So again, structure related to function. And what's happening in the model is during tape strips, you see a hyperproliferation, so an increase in the number and proportion of proliferative cells reacting to the insult. We can take our models, we can unclip them from the inserts, and we can have our skin then presented as a flat surface, which is very useful for these experiments where we're using commercial razors and studying the effect of shaving across the surface of human skin. And you can see the differences between dry shave versus the inclusion of a shaving gel. This is important for, of course, if you're interested in developing these sorts of consumer products. This is an assay which can be utilized. You can also injure the surface of the model through a mechanical blade. So we can do this. You can see at the bottom, day 0, very clearly a vertical scratch on the surface of the skin, which over time begins to recover. Some of these injuries can be just partial, just through the epidermis, often mimicking a nick by a razor, for example. And these are, as they are small injuries, they tend to repair relatively quickly over a week or so. And you can follow that quite nicely. You can see that in this example, how the wound first opens through contraction, then closes as the cells migrate in, proliferate. We can also look at burns. So this is a rig which we've developed with a branding iron, which at different temperatures can be used to brand the surface of the skin, so giving a burn. So these are 70-degree burns, and you can see how at day 0 the burn area is very wide, but it then begins to close and recover over a 2-week period. You can take measurements from this, and you can also see how the water loss from that area is also changing. We've also got machines for imaging directly from the surface, so you can do this in a live model in the presence and absence of different formulations to enhance wound repair. And you can look in detail, if you wish, at the various molecules expressed and regulated during the recovery. In this example, we've got a neurosensory model where we've introduced neurons. This is important for products which may have or give a consumer a burning sensation when they are first applied, and the developers want to understand this process. So we've incorporated sensory neurons into the human model. You can see them, the anatomy of this very clearly. And the idea being that if you stimulate then those neurons, they will release neuropeptides, which you can then assay for in the media. And that's what we've done here with an example, stimulating with capsaicin. We also have immune cells within our model. There are different types. We have different types of macrophage, M1, M2, also Langerhans cells in the epidermis as well. And this, of course, would be important for modeling inflammatory disorders. So that's the skin system. What we're going to do now is just briefly finish with the intestinal model. So the skin model is made with primary keratinocytes and fibroblasts, whereas the intestinal model is utilizing an existing cell line. So CACO2 is used commonly in industry, and it's often used in trans wells. The structure of this epithelium in this model does not bear resemblance to the structure of the epithelial cells in the real tissue. And there are other abnormalities with the existing technology. It's oversimplistic, but nonetheless it's used widely. So we hypothesized that if we took CACO2 cells and put them in an alternative growth environment with the signals from the supporting fibroblasts underneath, that this would enhance their structure and their function. And this indeed is what happens. The CACO2 cells now become columnar in nature with a prominent brush border, and you can see that very clearly compared to their 2D counterparts. They can be maintained for several weeks if necessary. You can see examples here from 2 to 7 weeks. This is a scanning electron microscopy showing the submucosal layer. These are the fibroblasts upon which we then see the CACO2 cells. You now see scanning electron micrographs at the surface of the CACO2, the cobblestone appearance. You can just make out the microvilli, which you can clearly see at high magnification, along with the glycocalyx. Transmission electron microscopy shows the microvilli and the apical junctions, and the basal area shows the nice examples of the trilaminar basement membrane. Again, we take the intestinal model and we compare it to real intestinal tissue for various markers, and these data are presented here, and they are also published. With this model, we can take the inserts, we can put them in between 2 chambers, and we can create a nushing chamber system to enable us to study the electrophysiology of the model and the transport of molecules across that tissue construct. So that's the rig that we've set up for that purpose. But the other thing we've done here is we've also introduced into the intestinal model immune cells. And we've done that to model inflammatory bowel disease, so IBD, ulcerative colitis and Crohn's. So IBD has essentially three of multiple pathological hallmarks, three of which we are prominent, and we can model these simultaneously in our system. So the first is obviously the immune cells and the effect of their signals. The second is the remodeling of the extracellular matrix, and the third is the integrity of the leaky gut. And I'll show you examples of all of these. So the first is in relation to incorporation of the immune cells into the model, and you can see the intestinal model here, and the CD68 staining of the immune cells is very apparent inside the system. So they're definitely there and they're present, and we can then stimulate them to release the cytokines. So we can do that in this case using LPS as one example of the stimulator, and then the profile of cytokines and chemokines produced from those models is similar to what you see in Crohn's and colitis. Under those circumstances as well, the stimulated release of the cytokines results in increased barrier permeability, so the leaky gut syndrome, and we can measure that using TEER, so that's trans epithelial electrical resistance, or we can add fluorescent dyes to the upper chamber and watch its passage through to the lower chamber, and do all these sorts of assays. We also see changes in the extracellular matrix within the model. So what was interesting here is that we saw in our model accumulations of MMP9 towards the epithelium. So we went back to the pathological tissue samples, and we then saw this unusual pattern of MMP9 expression in our in real tissue, and that showed us how and it demonstrated very nicely how the model can actually now allow us to identify new observations in the real tissue. So this particular model, the IBD system, can enable clients to test drugs and compounds incorporated into the medium and how they impact IBD. There are various endpoint assays depending upon what you're interested in, quantification of interleukin cytokines by Luminex and ELISA approaches, paracellular permeability, electrical transport studies, and of course looking at ECM structural components and their turnover through gene and protein expression assays. And we work very closely with the clients to develop the assays required for their needs. So in summary, bioengineering human tissues in vitro is based on this, for us, it's based on this physical support, Alvetex. We've also designed and manufactured our own plasticware to enable this to work effectively. Standing behind these systems are optimized SOPs. They are robust and reproducible systems. They're well characterized. They can be used for acute, chronic, and recovery studies. There is extensive data sets and applications which you can access through the REPROCELL website. There are multiple endpoint assays as demonstrated in this presentation and more. And as I already said, we work very closely with our clients to develop customized solutions for their needs. The last slide is just to point you towards the fact that you can purchase this technology from REPROCELL or its distributors, and there's a link for the plasticware there. But you can also engage with our sales teams and start to talk about service opportunities used through a CRO model, gaining access to these tissue systems. So I'd like to thank you for your attention.